Mike. Good morning, everybody. Welcome to ROV Sebastian Dive 954, live from Research Vessel Falcor 2. We've just got the vehicle in the water and we're just beginning our descent. We're going down around 1,100 meters today. So we're going to shut off the mics now and we'll come back on live with the audio when we get to the bottom.
Okay, and you're on in the corner then.
All right. I guess good morning. We just touched down to what kind of shark?
Baby shark.
Baby shark. Oh gosh.
Don't even know. Shush. You're not allowed to say the whole thing loud, okay?
No.
No. Just
Oh. Okay.
Yeah.
He's just waking up too.
Someone on the chat will tell us what topic
Yeah.
it is.
It's a good omen, I think.
Zoom in.
Stand still. Stop. Stop. Oh, beautiful.
Yeah.
Looks like one of those poor beagles. It looks angry at that.
It does.
It does look, it looks like it's frowning, huh?
It does look like it's frowning.
Baby shark.
Wait, do its left and right eye look different? No, it's just the light. Okay. Wow.
Okay, someone ID'd it, yeah.
Ray, is it a cat shark?
Cat shark.
All right.
Shark.
We'll see. Maybe.
Anyway.
All right.
Same thing.
Wait.
The white balance.
Yeah, I can find something. You do it. Keep going ahead.
Had a very angular head, whatever it was.
Yeah, that's why it's not a big water catcher.
All right. Good morning. We are at, what's our depth? What? 1,100 meters water depth. Just starting today's dive very early. Well, depending on where you are, I guess. Is it a strong current here? Or?
When we were coming down, it was pushing us, yeah.
Yeah. In the midwater or down?
Midwater.
Yeah.
I'll tell you in a minute if the current is strong.
Cool. Yeah, so this sediment looks a little bit more gravelly than it has been. Not just mud. And I'm commenting on it because the first thing we're going to do before we start heading towards some of the features that we mapped overnight.
I got pushed west.
Push west. Okay. We're going to do a heat flow probe measurement.
So 200 meters away from 1.
Okay. Are we? Oh, I see. Cool. It doesn't. We don't really care where it is as much as.
Just in sediment.
Yep. Yeah.
Yeah, looks okay.
Yeah. Why don't we try here? Because it might get rockier once we get closer.
Exactly.
Yep.
Yeah.
Cool. Alright. So do you want to do a bottom water calibration or?
I just have to. Sure. Is the probe on? Just turn the probe on. Okay, it's always on.
Okay. Power to the probe.
Bridge RV.
Okay.
And Tom, could you
Yeah.
come to
Yeah.
stop there? We're gonna take a heat flow measurement here.
Yeah, let's go.
Thank you.
Yep. Cool. We're gonna poke first.
Yeah. Just grab the
Yep.
So it says conduct sampling at seeps. Like, we're taking the
If
seeps
you
and muscles.
want-
Like, what does that mean?
you could look with that.
Um,
You might get a-
High priority.
I think I assume based on the hit list.
Which is weird.
Well, the same one that we've had.
But we've collected some of that sample.
Yeah.
We want more
Hmm.
muscles? What muscles is that?
Very soft. Good. Okay. Cool. We're ready to,
Yeah, I'm recording.
as long as we're,
Yeah, let's anchor down.
yeah. Can't tell if there's anything bright under the probe, but maybe it looks soft there too.
Okay.
Okay.
Probe going deep. Yeah, no problem.
All the way down. Awesome. Great. All right, so sorry to be quiet. We weren't actually talking. I assume you can hear me now. We are just landed at one of the sites on the accretionary prism in Trinidad and Tobago's waters. So this is yet another site that is the result of lots of stress and compression and compaction of sediments at the base of this pile that's been building up over millions of years. Looks like we've got some occupants of this carbonate mound in front of us. So we're in the process of collecting a heat flow measurement. We're just going to do one of these before we start heading up the slope of what is in an area called the Mud Province, named by some previous researchers based on the prevalence of lots of mud volcanoes in this area. Mud volcanoes are also I think there's over 20 in southern Trinidad as well. And these are common in this area because of this massive pile of sediment at the base of which you have really high pressure, and fluids interacting with sediments start behaving in ways that are similar to liquefied sediments. And as they're liquefied or their pressure increases, they become buoyant, and along with the methane and other gases associated with hydrocarbons, they make their way up along fractures and faults in the prism and exit the seafloor. And over many, many years, thousands of years, maybe millions of years, you end up with a volcano-like structure that is made up of mud. A little shrimp there. A couple sponges, lots of sponges, little coral, anemone. Yeah, so we're taking a heat flow measurement. You can't see the camera that we see, but there is a lance that is inserted into the sediments, and it's mounted on a track on the side of the sub. So first, before we send that into the sediment, we want to make sure that it's soft, and there isn't some hard crust just under the sediments. So make sure it's soft down to the depths we need to make a complete measurement. And then within that lance, there are 9 thermistors embedded within that titanium lance, and we're measuring temperature at each of those locations, those evenly spaced thermistors, and from that we will get a sense of how temperature is increasing as you go below the sediment. So we know the temperature of the water just above the sediment, and this gradient is part of what we need to calculate heat flow. So in this region, there's, I don't know how, dozens of mud volcanoes. This area was previously mapped, and those data were interpreted by some French researchers. Their data was not shared or made public, so we're working from published peer-reviewed papers and the figures without actually having the data. So overnight we spent some time mapping this area to decide where to focus our energy today, and in doing so found a number of really cool features. One of them, we're going to be heading up today, and saw some seeps as well in the data suggesting methane seepage, active methane seepage. So we're hoping to find some of the similar scenes that we've been looking for the past 3 weeks. Cool. There's another crab. Mm.
Are you finished?
No. Yeah, it takes, this one, it'll take 14 minutes total.
Okay. Yeah, I was trying to design a removal tool with just one lever,
Yeah.
and I would not have gotten it to you guys in time, so I just had to make the simplest thing I could.
No, it's great. Oh,
You're a bit low.
am I?
Yeah.
I've never been told I'm quiet. That's,
So do you think that tool could be used in any other research vessel? Is everyone's
yeah, they're all very
push flow
similar.
the same?
Yep.
Uh.
Yep, definitely. I mean, and that's that hard part, right? Is getting that piece open. Because you want it to be tight, but that makes it hard to
Yeah.
separate from the sample. This is a good sign when we see this much well, these little carbonate rocks and lots of life on the approach. Is that a sponge back there too? Yeah. What did we see on the way down? Polychaete?
Yeah,
Yeah.
Chris.
That was cool. Tomoctris? Okay.
I think that was on the Atlas.
Yeah. Tomoctris. And the Benthectinophore. Another one. Cool. So somebody earlier in the chat asked what our deepest dive was, and I think it was like 4,100, somewhere around there. And that was pretty far to the east of where we are now, in the abyss. And we're now back at shallower depths, but still, yeah, what's going on there? That wasn't us, right? Huh, who lives in there? Crab? Yeah.
Trying
Oh.
to find a hole on the left.
Yeah,
There's those thick deodorant.
those holes. What are those?
This is thick deodorant.
Okay.
Thick.
Hmm. How's it looking?
It's good. There's more fish in the deep than normal, so that's
Yeah,
just
that makes
sediments are
sense.
different.
Yeah.
We have about 5 minutes left.
Cool. Yeah, that may be a crab. Crab burrow. So we're gonna cross over this stretch of slightly rocky or rubbly, more sandy than muddy, which is interesting. It's different composition than we have been seeing. And head up the structure that was previously mapped as a mud volcano and looks like it may have collapsed a little bit based on its structure. Not clear, but based on the bubbles that were imaged on the multi-beam, this is another one of the cold seeps that we've spent the last 3 weeks investigating.
Mhm.
Mhm. We're done? Okay. Is the measurement done or no? 2 more minutes?
Yeah, we're at 5 minutes past the minutes.
Okay. All right.
Oops. Hold on. What did that? No.
I know. I'm just trying to get out of it.
No, I wanna move the RV.
Yes.
Or you wanna see if we could put it in the box, the rock box. I'm thinking about the other guys, they might have trouble taking it out from there. No, we can watch out and we'll get it out. It's not my bad. More donors stand by. Okay.
All right.
Okay. Heat flow coming up.
Yep. Thank you.
Okay, heat flow is up. 14 out. Is it enough?
Lovely.
Yeah. Just hold it down there. Bueno.
All right. We finished our heat flow measurement, so we're gonna head up to next to where we saw those seeps on the multi-beam overnight. So we climbing up onto this structure that looks, it looks complicated. Looks like maybe a caldera or some other structure associated with mud volcanism.
Keep the
Oh,
heat flow probe on or anyone.
we can turn it off. Yeah.
There's a rock coming up.
We'll just keep an eye on it. Oh, cool.
Oh, where the-
Whoa. Nice. Awesome.
You wanna look around?
Yeah. Just check this out on the way, Anna. Yeah.
Look at all this mud walls.
Yeah, there's a lot going on this massive carbonate mound here. So this is just covered in different corals.
What was that?
I'm trying to see if that is,
When we zoom in.
Zoom in there.
yeah,
Right?
just to see if it's,
Nope.
no,
Scale.
no, those are,
Zoom out.
Strange.
yeah, are those the same things that are alive up closer? No? No.
I don't know what that could be, 'cause it's a lattice,
No, it almost,
so
it's a lattice, but it's
Yeah.
not man-made.
No.
So this might be the one that Diva was talking about yesterday, that is that, can we get a picture of that, just to,
Coming after it.
yeah. Try and identify
Yeah.
what it was. It's no longer alive, but
It's very dead.
very, very dead. Huh. Get a picture of that.
Okay, zoom in again.
Yeah. Huh.
Yeah, it's on the look of that one.
Cool.
Move on, or you still
Yeah.
want to look at
We
Yeah,
can-
I need to zoom in a bit more.
Yeah. Just to,
And just fix on it, but without moving if possible.
it? I'll have the camera stabilize your RV.
Yeah, please.
yeah, there's like a brittle star under that dead coral, I assume. Yeah, so look, right down here, there's a living example of it.
Oh,
Yep.
Oh, I don't know if it's
Well, more likely than the other one. Huh.
That's good.
Yeah.
Oh, that looks like that looks like it's a dead skeleton of a
Yeah, honeycomb.
reptile, maybe. It's a stony coral skeleton.
Okay. Cool.
Sit down, zoom on.
Oh yeah, that looks better.
Normally we have all of those on.
Hmm.
Gyres.
Okay. So these are normally on. Yeah.
It's good.
Okay. Good. All right. Head back up towards the
Let's start.
waypoint. Yeah. At the start, yeah, exactly. The first waypoint.
So,
Yeah.
It says stony coral skeleton.
Cool. Yeah, this is a good example of how any hard surface that's available at the seafloor is an ideal place for all of the things we're seeing here that are, sessile creatures that need a place to attach to. So even in the abyss, some of those rocks or nodules can provide habitat.
Yeah. See it.
I don't know. It's another one of those whatchamacallits from yesterday.
Straight into type
Oh,
of
man.
fish.
Yeah, no, I know, but what's it called? What was it called? Telescope fish? It had two different size? It might have been a telescope fish. We saw one yesterday.
There's something on the left that you want to look at that, or head.
Yeah. No, I mean, we can explore around these.
There's something big on the left.
Yeah. There's a cool coral up ahead. Whoa. What color is that? Is that purple? Whoa. Can we take a look at that, please?
Yeah, I think we
This-
will have
yeah,
a piece of this.
We need a piece of this. Wow. It's got a sea star on it. Probably
Brittle star.
munching on it. Brittle star and multiple brittle stars.
Yeah, if we could do some good video.
Yeah, this is beautiful. God.
Get that this
So cool. We definitely need some good video of this. That is so beautiful. Wow.
Yeah, that's right. Yeah, so they want good videos and pictures of this and sample.
Cool. Is that sea star munching on that? Or is that just an enormous...
No, it's a brittle star. So the brittle
That
stars
is the
don't need-
fattest brittle star I've ever seen.
So they don't need coral. They just use the coral.
Okay, but why is it so big?
Species.
Yeah, it was
Just huge. Yeah.
If you walk out, you'll see it flying out right
Right, yeah, the sea stars that were actually eating the coral, but the brittle stars.
Yeah, this
use
one's
it
just
to go
hanging
up
out.
so
Yeah.
that they can
Yeah.
fire it.
I've just never seen a brittle star this fat. I mean, big boned. I did not. And look at that that base of that coral is just giant. Okay, we'll hang out here to get some glamour shots once it settles down.
I just hope the current is not behind us. Because if the current is coming from behind us, we're gonna need to change sides.
You will what?
If the current is coming from behind us,
Yeah.
never gonna change. Then we need to change.
But the current's east to west.
The current is east to west.
No.
Yeah, so it's like so beautiful.
Oh. I go behind Rachel.
Oh, you are? Yeah.
You wanna take a
No,
look?
the base of the coral is large, and also the brittle star is a different species that I'm used to seeing. It just looks, it's a big boned. And you're on. Okay. Animataxis is replacing me. Have a lovely morning.
It's not working.
Yes, it is. It's 1 a.m. It's morning to these people. It's morning to me.
Okay, good morning. Good afternoon, good evening, wherever you are watching us from. My name is Anna Metaxas. I'm from Canada. And we're here on this cruise in Trinidad and Tobago waters. We have been traveling in this area and exploring and collecting samples for the last 3 weeks, and we're on our last week. And we have been really quite successful, particularly in locating methane seeps, which was one of the goals. And you just heard from Dr. Rachel Lauer, who's been, along with Robert Perrin, critical in actually locating those seeps in this vast ocean by multi-beaming and mapping essentially where bubbles of methane that escape from the seabed are found in the water column. So this is another site where we did see bubbles during mapping, and so we're hoping to find bubbles actually quite soon once we move up this hill. But in the process, we're sampling this beautiful octocoral that you see in front of us. We've only seen this on a couple of other occasions on a previous dive. So yeah, we will collect a piece so that we can bring it on board and do some genetic analysis, identify the species, etc., etc. So first we started by taking some nice pictures of the coral in situ, so in place, so we can get the coral and the color and the behavior. We're watching the polyps that are used for feeding, capturing food, and then we will collect a small sample. Sure. Can we also have lasers on? I'm sorry, I can't see them. I don't know if they're on already, but.
Yeah, they are on.
Oh, yeah, they're at the bottom there. I see them now. Yes. Okay, so if we could zoom in just enough to have both lasers. Why do I only see one? Where's the other one? Yeah, maybe because we need those lasers. We know the distance between the lasers is 10 centimeters, and we can use them as a scale to know how big this coral is. But I seem to be unable to see them this morning. Perhaps it's because it's 1:30 here, 2 o'clock. Can you? Okay, here we go. There's one. Is it the angle or the light? Or?
I can see both of them. I got
Can you see
pictures.
both of them?
See
Perfect.
both.
Okay. As long as the pictures have it. Okay, great.
Got it.
So, yeah, Mike, if we could get a piece of this coral, put it in a quiver, please.
Can we get that branch sticking up or not? Because it's easier for them.
Sure. That's fine. You can take that branch. Yes. So, the thing about these corals is they'll grow back. And so, we will take as small a piece as possible. That'd be sufficient for our sampling. But you know, it's okay. The corals grow back. They're colonial organisms. Each one of those little polyps that you see are individual of this colony, very much like shallow water corals. And so they just divide and divide and divide, and that's how this coral grows. What you see also in this coral is this very large brittle stars. So we have different types of animals that use the coral for different reasons. So a few dives back, I think it was 2 dives ago now, what we saw was a sea star, which was a relative of the brittle star, actually feed on the coral itself. Now what we see here is different. We see a brittle star that has climbed up to the coral because there's a lot more current here. And so because it's this higher up, it can actually capture food from the water as it's floating by. And so you can see it's moving its arms into the water. And the reason it's doing that is to capture particles, which it then transports into its stomach with little hair-like cilia and consumes them. So here we have the coral is used as a structure to support this animal and not as food itself.
Do you want to put a quiver or the bio-box?
Quiver, please. So this is quite interesting because we already passed some large boulders along the way very early on this dive. And what is otherwise a very sort of sandy, muddy bottom, soft sedimented. And so there's a lot of studies now that are starting to look at the importance of those boulders. So an animal like the coral that we see here can only live on hard rock. It can't attach into the sediment. It needs hard rock to attach. And you see it has a very large holdfast with which it has attached onto this rock. And so having those rocks interspersed along the way can provide stepping stones for those animals to disperse from one place to another. Otherwise, they would have these vast areas of solid sediment where they wouldn't find a place that was suitable for them to attach and grow. Um, and so those rocks are really quite important stepping stones for the species to be able to disperse from one location to another. Mike, but
What's a bigger?
I can't see the stick. So, but this big.
What size? Yeah, no, I just.
You know the size.
I cannot reach for put to the blade.
Oh yeah.
My limb.
Yeah,
I
yeah.
Go a little bit further.
Yeah, so you can see the polyps have closed up now. They're disturbed by it because they felt the pressure of the arm. So you can see the ones lower down, which haven't really felt it. They're a little bit more open, but the ones at the top where we were trying to sample are closed. So you can see in the background the arms of the brittle star just waving in the current, looking for food.
Não tá travado no chão, né? Coloque. Um monte de textura ali, você não tá se mexendo. Vai, yeah, that's good. Stop.
Every night I bring the pens in, and every night they disappear. I know. We have reposition. Lower.
This size.
Lower, lower, man.
A little lower.
Lower.
A little. Yeah, that should do it, Mike. Yep, perfect.
You have more shots.
Yes, please. Zoom in. So you can see the polyps are now completely closed, so all those beautiful purple polyps are disappeared. Nice, thank you. Sample number?
It is 467. 467.
It's good, thank you. It's going into sorry, 467. I'm still asleep, so I'll give it to me.
Do you need it more, Anna? Anna? Do
Sorry?
you need it more, or is it okay?
No,
May I close that one?
yeah, you may. Yeah, sure. Perfect. Thank you. So now that we've collected our sample, we will continue moving up towards those red the first set of bubbles. Can we put the bubbles on the screen? On the as waypoints?
But they're up there.
The red,
Yeah, the red ones are there.
yeah, if we could put them as waypoints, did we?
It is.
Okay, they are perfect. Yeah, we had them yesterday. So, yeah, so we're heading towards that first set of bubbles that you see. I think it's the blue. Are they the? Which ones are they?
The first waypoint.
We don't need to go to the first waypoint. We need to go to the first set of bubbles.
This one?
It looks closer to one than two.
The blue ones are.
Bubbles?
Yeah.
All the blue ones, okay.
So we'll head there.
Marcelo, let's see.
Which
So we can come here. This is one.
are those?
Closest one is this.
Okay. Wow. Looks very different on the filter mouse.
I think on Peter Mouse it's in the water column.
Oh, that's why. Yeah. Okay. So I guess let's head there slowly. So again, we see today we started at a much sort of rockier site than we did yesterday, and most of our dives usually because we started up the flank of this structure as opposed to down on the bottom. So we'll see a lot more large boulders and gravel in the sediment. And so our first, our first target is a location where we have seen bubbles in the water
Please,
column.
Move towards the proper waypoint, please.
Yeah, it's going to be waypoint side of point.
Okay, thanks, Tom.
So as we pass, we see this beautiful little stars and feeding up in the water column. We see black corals. Some sea bands in the sediment. And a lot more rock. You can see we're at a bit higher, we're flying a little bit higher than usual, and that is because we're actually at an angle, and so we need to keep the RV off scraping the bottom. Wow, it's another large boulder that we're approaching, and we have corals here, basket stars, brittle stars. Corals on the top, very similar to what we were just seeing earlier on. can see a skeleton of a bamboo coral lying on the seabed there. Crinoid. It's a blue sponge. Do you think they will want a blue sponge? You see it. Maybe if we see it again, we'll stop.
Okay.
Another one of those purple corals. It's this site 6 and previous dive and this dive here is the first time that we're actually seeing branching stony corals. Flabellum is the stony corals that we see like in the sediment or attached onto the rocks, but they're small. They don't create the slightest structures, but we have seen both in the previous and this dive stony corals. So these are deep sea stony corals that can form large reefs. We haven't seen large reefs here, but are known to form large reefs in other parts of the world. So it's so interesting to see the different places that would appear to be similar in terms of the environment, and not too far from one another, can have different species from one another. How come is this because of the particle in the water that it's focusing in and out? Like it's sort of the focus
I
just.
can take out focus out.
Yeah, maybe, because then we can sort of, you guys can control it. So our dives have been getting shallower as we're progressing nearer to the continental shelf, and eventually we'll do some dives in mesophotic reefs. So we have some very, we've done some deep dives at 4,100 meters in the abyss and beyond the exclusive economic zone of Trinidad and Tobago. And so now we're back in the exclusive economic zone, and we've seen many different types of habitats, of course, depending both on depth but also on sort of geological history and mechanisms of formation. There's a big coral coming up. There's a little bit on a boulder. Yeah.
What's this star? Is that a starfish?
It's a brittle star, really.
Oh, brittle star.
Yeah. Can we take a look at the coral, please? I don't know yet. We could zoom in, please. It's an octocoral. Octocoral, yeah.
Okay.
Sorry, you were asking about the big
Yeah.
things, not the, yeah, those are sea stars, they're Brazingid sea
Oh,
stars.
okay. Sea stars.
So yeah, if we could zoom in a bit, please. Maybe we should sit down so we can have a nice imagery. No,
What do you, Jeremy?
it seems hard to focus when flying over.
We're gonna stop a moment.
Okay, go ahead, stop.
Thank you.
Sorry?
Stitching there, so.
Oh, I
try a
see.
different angle.
Yeah, okay. Yeah. Tricky. So yes, there's a comment about the long arms of the spot lobsters that live on the coral. Yes, they are long, so they can grab food out of the water column. In fact, yesterday, I believe, we actually caught one of them feeding on a. It was actually a ctenophore, which is an attached, a benthic ctenophore that was attached further down on the coral, and so they had grabbed it and went feeding on it. Could you zoom without landing, like relative stability? Or?
We're gonna change the other side to the probe on the deepest side.
Yep.
Because the probe on the shallow side, you know,
Yeah.
that's any rock.
It gets stuck.
And you're gonna be hit.
Hits the bum hits. So this is a bit tricky again because we're on an angle. We're on a slope here, and so it's we're trying to take a good look at this coral, but because of the angle of the seabed, it's difficult for the sub to land and not be able to be flat as opposed to hit the sediment, which seems to be a bit tricky here. Hmm. Yeah, maybe we should just avoid. It looks messy. Yeah, because then it's gonna be this. Okay. Well, we can try. Okay, that. I think you can zoom in the axle. We can. Oh. Ha! Landed. Almost. There we are. Beautiful. Oh, look, there's a polygate worm there as an associate. The other side, we saw a lot of 2 or 3 squad lobsters. You see the polyps that are sticking out to feet. Oh, and an arm. And then this one's in the front. Oh, look at there's the arm from the back. Just focus on them. On the ones in the front, on the front. We've seen the back, so that's great. So let's look at the front too as well. Just, yeah, it's here. Perfect. Yeah. Nice. Yeah, that's great. Thank you. Carry on when you're ready.
Anything else, Nina?
No, that's good. Thank you. Why do those corals grow up to one side only? Yes, that's an interesting question. So, because the corals are suspension feeders, what this means is the grab particles that are suspended in the water. Often, what we see is that the orientation is in the dominant direction of the current, and so what that does is it maximizes the probability that they will capture food because the entire fan is opened up in the dominant direction. Now the current can change, but most of the time it's running in this direction that you see the corals oriented in. Often, or sometimes, some species what they do, they even curve into the current, and so that even increases even more the probability that they're going to capture food because it's trapped. It gets trapped into the structure of the fan. So we're carrying on towards the first set of bubbles. First set being the one lowest first on our track that we have mapped, based on the mapping that our trusted geophysicists have done overnight. Well, the ship does the mapping and the geophysicists interpret it. So we're very fortunate to have this ability to do the mapping, because what we do is we'll arrive at the site and the ship will run this grid along the seabed where they basically map the bathymetry, so the depths of the seabed, but at the same time they have a piece of equipment that can identify bubbles in the water. And so then that's when our geophysicists get on and working on to trying to interpret where those bubbles are and if they would be a good place to dive, giving us a good chance of seeing methane seeps. And so far, they have been pretty accurate. So a big shout out to Rachel Lauer and Robert Perrin from University of Calgary for doing this work for us. So interestingly here, we're not seeing yet evidence of animals that live on seeps. So on other dives, when we start approaching these areas of bubbles, we start seeing, mussel shells or dead tube worms, etc. So we haven't seen those yet. I expect we will as we get closer to the seep. The thing about it is, because the deep sea is so poorly explored, I mean, we have seen less than 5% or not seen, but sampled less than 5% of Trinidadian deep water. You never know what you will find. We kind of have some expectation once we've done a few dives, but then always something surprising turns up, which is what is great about exploring, having this opportunity to explore these areas and these amazing tools to do it with. See quite a few of those precision sea stars. Here's another one of those black corals. We saw one of those yesterday and we did sample it, so we don't need to We did sample it, didn't we? Maybe we didn't. I'm not sure whether we did, so maybe we should keep going. Yeah. We'll keep going, yeah. Thank you.
Pretty dirty.
RV Bridge?
Good morning. Didn't move your waypoint.
I'll take waypoint.
Huh. Getting there. Getting there. Oh. Bubbles turned yellow. Oh, this one. So again, we're on the slope for those of you that are joining now. We're on the slope of a structure that we hope has some methane seeps, particularly near the top. But there is a site that we're heading to now, which is we recorded some bubbles earlier on during mapping at night. So we're almost there. About 30 meters away.
They got the flight events map on the screen, so they can sort of follow along with you.
And we'll check that out, and then depending on what we see, if we see seeps there, then we'll stop and take a look. If not, we'll carry on up the hill, because at the top there was quite a few hits with bubbles, so we expect to definitely be able to locate methane seeps at the top of this hill, which is depth is 20.3. So that would be about maybe 80 meters shallower than where we are now. Yeah.
Seems like there's more stuff this way.
It's some sort of harder stuff. So, but it could be just the rocks, you know. So there's so many rocks here. So what we're doing is we're seeing some hard targets on the sonar. We suggest that, as we're seeing, there's a lot rock, and so we see the rock to our right hand side here. But usually we get very excited about rocks because we're on the flat and it's sandy. But here we've been seeing quite a few of those large rocks that seem to not have evidence of seepage. Most of all the animals on them are animals that live in non-seep environments like corals. But what is those white? You see those right there? Those could be tube worms. That rock, I can't tell. That stuff there, if we could zoom into. Just zoom in, we don't need to sit down, just to see if there are tube worms. Yes, they are. Okay, so there's a beautiful black coral going by, but this is the first tube worms that we see. So the first evidence on this dive that there must be some sort of seepage somewhere. These animals live close to seepage areas. Yeah, we're gonna, I guess, turn and try to go by those bubbles and see what we see. Here's a nice black coral on this rock. So, yeah, just head to 06, whatever that is, the bubbles. Up the hill we go. That's strong signals. And
The ones on the side, they were a little bit weaker. And then the ones on top, those
right. There are some benthic ctenophores on that stalk that we saw there passing. There's quite a few that we see here, the stalks, all those little red fuzzy bits on the stalks. These are all benthic ctenophores. Oh, there's a brittle star running away. So we're just gonna slowly make our way up the hill in search of methane
Your
seeps.
waypoint, please.
Okay, wait.
Is that a clown? No, don't worry about it. Keep going. So these particles that you see in the water, it's called marine snow, and this is all material that is raining down from above. This is sort of dead phytoplankton, dead zooplankton that are actually, except for methane seeps that have their own system of producing carbon, this is the main source of food for the animals that live in the deep sea, and that's why it's hard to make a living because the food is limited down here, and so you don't see large dense populations of animals, right? So you see a coral here, a coral there, but you don't see a lot of animals, at least those large animals that live on the surface of the sediment. There are quite a few animals that you don't see that are below the surface of the sediment. We see evidence of those animals either because to make holes or burrows or mounds that we see on the seabed, and they take advantage of that particulate, those particles that rain on the seabed from above. Now, methane seeps are different, and so when we get to the methane seeps, you will see that there's a lot of animals there. There's a lot of mussels in particular. There's sponges, there are tube worms, and the reason for that is because production of food, of organic carbon, is local. It happens at the seep by microbes, which replace sort of plants as the primary producers in this particular environment. And so because they use the chemicals from the seep to grow on, then they are a source of food, the base of the food chain. And because there's a lot of methane that comes out of those seeps, then you can have large populations of those microbes that then in turn support all those other animals. So we will see a big change, right? That's the big thing, the big difference between the communities that are away from seeps and not from the influence of seeps, and then and the communities at seeps that have a lot more biomass, a lot more animals. Starting to see clam shells, I think. Yep. Or mussel shells. Very few, but we're starting to see them. So this is a good sign because that means that there is a seep up there. So I think I'm gonna sign off now as I leave you climbing up the hill towards the first sign of a seep, and my colleague Jamie will come on and will take you further up the hill. Thank you.
Across the red dot. Yeah. So they're all put in the Yeah. And it's been very different because we started on the hill, so it's perfect. Yeah, it's good.
Yeah.
here? Yeah. Sorry. Good morning, everyone. My name is Jamie. I am an MPhil student at the University of the West Indies studying deep sea biology. Let's keep climbing this hill and find some seeps. Oh, that's a big shrimp.
Over 2 meters.
Yeah, with all the rocks. So we detected some bubble streams while mapping last night, but they haven't been visually confirmed. So as we move up this mound, we're making our way to all the different points that indicated there might be seepage and looking for any high density communities or biodiversity hotspots.
Is there a collection of shells over there on the rock?
If we've collected shell,
No, is that a collection of shells? Clam shells there.
the white one?
Yeah.
Yeah, those are dead shells, dead old clam shells. Oh, fish. Crying. Yeah, these are like shell aggregates, so it shows that there is a seep somewhere, or at least there was a seep nearby.
Bridge, RV.
RV bridge?
Maybe slow down a bit.
Yep, reducing the speed.
Thank you.
Yeah, at the top of the mountain is where all the bubbles, all the seepage is. lot of cup corals and fish.
White coral that we're seeing is of coral. It's not.
No.
I just put it as a
Coral.
coral.
That's fine. The octocorals, they don't have like any.
Really like
Yeah.
each one.
They're sponge. Yeah.
What are you, Jovi?
Are we able to look down?
RV bridge?
You resume normal speed, please.
Yep, coming back to 6.5.
Oh, that's a big crab. The big corals coming up. Yeah. Down there. Oh, this is beautiful. Wow. Probably want to get good imagery of this. You probably want to get good video of the different corals. Yeah.
Wow.
Yeah.
Oh wow.
Oh, look at that coral. There's a pencil urchin.
What is that blue thing?
The blue?
Yeah, you see it?
Maybe a
Some-
sponge. Sponge is always my default. Wow, look at that bird. brittle stuff.
Should we?
Should we what?
Tell Usha.
I guess we're good. Hold on.
We can. Yeah, we can land for a minute. I think.
Yeah.
Land for a minute. We'll get good video with zooms.
Bridge, are okay?
Species of science gives contact coral gardens.
Go ahead, orbit.
Sorry, buddy, we're gonna land here. Can you stop?
Yeah, come to stop, Mark.
What's the phone number?
Are you?
Excellent.
I can do it.
Yeah. What's her phone number? Do we know?
I just took it from you.
Yeah.
That little squat lobster.
Thanks. I'm calling from the other. You're not calling. Oh, you're calling on
Oh yeah.
try that. Give me. You're calling her on WhatsApp.
Yeah. If not, I'm close to this.
Yeah. Or from the other.
Who are you getting rid of? BBC? Usha's left the number now.
Yes, the Raven numbers they're not running up to the
Can we zoom in on that? Oh, where?
That's her cabin number. That's
Cisco
not-
place?
the
Just
phone.
everything, I guess.
Is the phone the same as the cabin
Not very
number?
specific.
1380.
The
Oh.
squat lobsters.
Oh, sorry, Jim.
Yeah.
Thanks, Jimbo. Is it supposed to be ringing? Oh, it's ringing now.
I hope so.
Look at them. Little amphipods. Oh. There we go.
You're not answering?
Hello? Anyone missing some
Oh, look at that. Perfect. Yeah. Bright colored.
She said she's gonna take a look at the screen and digest it.
Can we zoom out so I show her? Zoom out so she can see us
Realistic.
close.
Yeah, I don't know which one's more realistic, but I like the one at the bottom.
I can look around.
This is so cool.
Move to the left. See what they're in. There's the restroom. And there's a lot in the back as well.
Yeah.
We'll get to- Look at the amount of brittle stars.
Look at all those brittle stars.
Yeah.
They said
Zoom in
they're
on that.
okay.
They're on their way.
Want it?
Yeah.
Who did they say this to?
Hmm?
Did it come in the?
She sent a message. She said, back to me, very nice, but we out with you.
Okay. Good. This girl there they're gonna get. But it's fine.
They don't want it.
No, they don't want it. Good.
Well, at least they saw.
More for us.
Yeah.
So they're not.
No.
Okay, cool.
So, yeah, let's look around.
Let's look around a little
This is
bit.
Pretty.
Yeah.
Yeah, let's take some more video. You know, it would be good if we could get some of those for us with the lasers on, so we can
get sizes.
Yes,
This
sounds
one
good.
is huge. In front of us.
Maybe stay a little bit longer without zooming in.
Yes.
Okay. That? What? What? It looks like an interesting
Yeah, so can we like
squat
not,
lobster. What is that, Alan?
what is what? I can't, you have to stop moving.
Sorry. That.
Wow. The spiky thing.
Yeah. I mean,
What do you mean? The spot lobster in front of us.
Yeah. But
just look. He just says spiky. Okay.
yeah, the spiky one.
I don't know.
Oh,
Okay. So let's zoom out
yeah, but
so that we can look at it
particular
with the leaves.
species.
Oh, gosh, I don't know. It's spot lobster.
And then we'll hold it there for a bit. Yeah, I can see them.
Bringing goodies.
Most of the way to the top of the hill, heading towards seafloor.
Zoom out for it.
Come across this amazing bunch of corals.
Lots of spot lobsters.
Videos, that's exactly stuff. Alright.
I think it's a
Yeah.
think. Or the pollets.
Specifically with the pH sensor, I'm going to look at the
Hold it for a second. Yeah. I think it's smaller. Corals.
The one, the
Purple?
blue, the
The
purple,
purple.
yeah, that's the one we sampled.
Yeah.
Yes, I'm going to work on the okay stuff.
Here's the dead part of that one. Look, there's
Yeah.
a little barnacle up at
Barnacle.
the top. Yeah, so, yeah, when we zoom out, put the
Put the
leaves
leaves
on
on.
the thing.
When we see the
And then-
whole.
Yeah,
And then explore all the ones behind.
yeah.
Oh. It reminds me of an orchid or a cherry blossom.
Yeah.
Alright. Let's zoom out.
Like a fish, man.
See the whole thing. Are the lasers on or they're just below? Ah. Oh no, yeah, I see them. Kind of on the screen. They're like in the lower middle.
Oh.
Yeah, sorry.
Okay, on the coral. Okay. Yeah, so that's 10.
Yeah, that's 10. That's what's up. You can wait there.
Coral.
You want the whole coral? Okay, zoom back
Yes,
out.
so we can measure.
Perfect, let's
Without
stay
cutting
there.
the top, without cutting the top.
Let's make sure the two green
So
dots
it's
are on.
about a meter high by probably a meter wide.
Yeah.
Yeah, that's great. Thanks.
Okay, cool. Let's look at the ones behind. We might have to fly. Yeah, the ones behind. Yeah, the one behind it. Wow.
Yeah.
I'm thinking we can maybe like we can maybe park down like over there so that we can probably look at so that we can look at like all the ones up on the rock. Are you able to?
No, I just go out the other way. Just
Yeah.
to turn my turn.
Mm.
Look at all the ones on the map. Yeah, we should.
Yeah, that's where we're going.
Yeah.
So we're gonna park behind so that we can
Yeah,
zoom in
yeah.
on all of
Yeah,
them.
for sure. Okay. I think so. Maybe coral and it's a coral leaf. This one is a hard coral. Stony coral right here. So you see the stony coral and the soft coral? We have a zigzag.
Mm-hmm.
So that's a stony coral, not the soft coral.
Mm. Okay.
Oh, like a big crab.
Oh wow.
Oh, yeah.
There's some massive crab. Does it have eggs?
Can we zoom in on the crab down there?
Okay, look.
Yeah.
Like its belly? Crab belly? Yes, two girls. Okay, she does.
So we want to zoom on that one.
I want to zoom on that orange bit below her with the book.
Just stand by. Just
Yeah.
not in good position here.
You know. Mm.
With rocks and everything.
Oh, okay.
Just
Yeah.
to balance.
You see this? So complex.
Wow. Is it? Those are eggs?
Yep.
Wow.
Eggs?
Mama crab. Let's just hold it there for a sec.
Those are all her eggs.
Wow.
Big mama.
Lots.
Lots and lots of eggs.
Yep. So the females typically stay out of the way and not move much when they have eggs.
To protect them.
Yeah.
Oh, wait, but can we focus on it? Refocus on it. There we go. Oh.
There must be like 5 billion eggs. Chance of survival of one.
We could probably.
Yeah.
Thank you. We'll probably zoom out now. Yeah. Now we want to turn back to look at the corals over there. Yeah, that one. Like on that rock face. We're just looking around the area.
Follow the line, let me know to move the best.
It's a massive shrimp.
Yeah.
Yeah, we can keep going and looking at those.
around a bit.
A little bit.
Put a waypoint there, corals.
Point here.
Waypoint here, yeah. Coral garden.
So should I keep going or?
Yeah, I'm just looking around.
Garden?
Yeah.
Okay, following that direction, I need to know to move the vessel.
We
If we keep going up on this
will have to
line,
move a little bit. Yeah.
so
I mean, I guess you can move it a little bit. I mean, we might park around here just to look around.
I will put it.
Okay. Just to look at those corals for a bit. Yeah, somewhere in the middle.
Maybe we'll just.
Soon by. Yeah. You can move the vessel because we'll probably won't
We're
walk
in
it.
zero view.
Just looking at it.
Go ahead, orbit.
Maybe start to move towards the waypoint, please.
Yeah, continue to move.
Thanks, mate.
Oh, it keeps going. Yeah.
There's a photographer who was living in the garden.
Yeah.
He's sober.
Wow.
So these are like 100 years old.
100?
Decades to 100.
You can see some dead ones too.
You see there's also quite a few skeletons.
Yeah.
Yeah.
Laying on the ground in between.
Yeah. I mean, some of them, look how thick that one is. That's an old coral line flat.
Yeah. That one? Yeah. Pink. Wow.
And they're all facing the same direction.
They're all facing the same way.
Ah.
Yeah.
Yeah.
Bombshells, aren't they?
Yeah.
What's over in the distance there?
We're
You know
moving
the
over
white
here.
stuff?
Yeah. We're heading there.
Great.
I'm wondering if it's just massive bacterial mats. Charlotte, I just realized we haven't taken any pictures of the coral garden to send in the group. Have you?
I did.
No, just for like later on when they wake up. No one knows.
No, I'm just.
Yeah. I'll send
Oh yeah, let's zoom in a little bit. We can zoom out. Yeah, we can keep going. We're just gonna explore and look around the area, see if we see any bubbles or any live mussels.
But obviously, yeah.
Yeah, there
Yeah.
was something.
It's up there.
Yeah. Straight to exactly where it's on the map. Where it seems to be. Wow.
Yeah.
Look at the amount of shells. No, I kind of just assume that this is old. Cyanide. move like this way probably. So we're just gonna explore like the whole top of this area, just because all the red boxes are types of bubbles. So.
Are we going to the top of the hill?
Yeah. Yes, we're gonna go to the top of the hill by waypoint 4, but we're just gonna look around the whole area.
What are you, Jeremy?
RV Rich?
I didn't move your waypoint. Good morning.
Oh, it's still Tom. I will move it.
Oh, okay, Tom. Thanks. Sorry.
We have some more shells over there. Just look. Treasure hunt. Trying to find the book. That looks like a coral. Yeah. Spot lobster. To go to waypoint 4? Yeah. Could be passed. this way a little bit. Just want to see. Yeah, we can keep going. We passed waypoint 3? Yeah. Yeah, long time. We were going through all the red ball, looking at all the
No, but on the
So there's, I mean, if you look at it, it has
Nice.
2 waypoints down there and up there. But
The blues are the bubbles, I think.
Yeah,
no
the
blues,
blue,
oh,
the
yeah.
blues don't mine here.
Yeah.
The one's supposed to
but we
be.
seem to be going in between.
okay. Keep going right. Yeah.
4, 5. These are green. 5 points up there. I don't know why. Which one?
See, yeah, you see? Yeah. The white thing there? Yeah. Still want to go back? Oh yeah, that's something we don't know about. Is this a mistake? No, that is.
No, those are bubbles.
Yeah, yeah, but they have two tracks.
Yellow is your
Yellow is
waypoint.
seepage. Yeah, but they're full like you do one right now.
They shouldn't be because this is okay.
So I should look at this map instead.
Those have been dragged or something's happened.
Yeah, that was
Something's been moved.
Could you tell me where else it should be?
Six is way back.
Same.
You want 6, yeah?
I think we still need to hit
No, we need to do all the.
You haven't done 4 and you haven't done 5 yet.
No, we just went on 5.
From the chart, we've done 3, 5, so just go up to the next one.
No, that's what I'm saying.
Yeah, he's just trying to find the next one.
No, I know it.
It's up there, it's the blue one.
There's
It's
6
not,
down, yeah.
no, but we've done 6, we've been there, so.
It came from there.
Yes,
It came from
we
there.
have, we've done those bubbles.
So,
So,
7.
we're coming
4.
from the bottom, right?
4.
So I don't know what the number is. So we've come from there, so we can see the track, we've been to 2, 3, 5, so then our next one is.
Alright.
You talking about the bubbles or the waypoints? We're getting a bit of confusion happening here.
They've
So-
got 3 bubbles.
You're talking about the bubbles, yeah? The blues.
Well, the
Blues
bubbles
are the
are
bubbles.
waypointed, right? So.
No, the yellows are actual physical waypoints and the bubbles come in as waypoints, but they're blue.
The blue text.
It automatically
But
knows.
it says waypoint T004.
There's no other way to display them on
Yeah,
there.
that's fine, that's, so I'm just, so,
There's
done
waypoint
5,
4.
so then we can go to 04, and then we go to 7.
As far as waypoints, 04 is on the top left of the screen in yellow. That's the
Yeah,
physical
so let's
waypoint.
just go.
Okay.
Sorry,
Go
I was
starboard
a bit confused.
and comes it up.
Yeah, no, that's right. So yellow is your waypoints.
I
blue
got
is
you.
bubbles. Just forget about the numbers of the bubbles.
Yeah, forget the
And
numbers. Just go
Yellow
from blue
is,
to blue.
yellow is your waypoints.
Alright.
So
Not
ask me
confusing
if
at
you
all.
need to know where your
Not-
waypoints
not
are. Okay,
confusing
we've
at
got
all.
4 people telling you different things.
Just go from blue to blue, and but in between yellow, like actual waypoints, like follow the blue.
Thank you. Alright, good morning.
How are you? Good, good, man.
rest.
Yeah, that's bad.
Let's go find
Good morning, everyone. My name is Kyle Foster, and I am a postgraduate student at the University of the West Indies St. Augustine campus, studying deep sea biology. And this morning, we are searching for bubbles, methane seeps. We're exploring a caldera-like mound that has possibly collapsed, and we're keeping our eyes peeled for any signs of methane seepage. So far, we've come across a quite interesting coral garden, so it's been an exciting dive, and yeah, I'm sure we'll see many more interesting things. Nice sea star over there. Almost looks like a sponge. Yeah. Sponge star?
Sponge star. SpongeBob, I'm touching a child.
And then if you're wondering just how large these organisms are or the environments that we're looking at, we have two green dots, which are laser projections, and they're spaced 10 centimeters apart. So they allow us to get an estimation of the size of these organisms. So we're seeing some brittle stars, lots of corals just dotted around on the rocks. The rocks provide a nice hard substrate for the corals to colonize. And so throughout this expedition, we've been mapping the seafloor using an instrument known as the multi-beam echo sounder. And what happens is it emits sound waves to the seafloor, and those waves are bounced back, and they help to paint a picture of the seafloor. So we're able to see mounds, valleys, channels, And this is nice. And associated with these different geological features would be different types of biodiversity. So that's how we choose where we want to explore. Lots of nice corals on this rock outcrop here. I think might have seen this purple type before. Lots of corals in this area. Maybe get a little zoom in of this one.
This one?
Yeah, just a slight. Got quite a large brittle star wrapped around this. Oh, quite a few. Another one. Gorgeous views this morning. Are we then going to turn left and head to the next loop? We can head to what's marked as 7.
Roger RV, update to waypoint.
It's a big rock. It's nice as well. All these over on, I mean, it's like the same type of coral, I think, but lots of big brittle stars on it as well. Yes. And this one has some anemones on it. I personally don't think I've seen brittle stars these
That's
these
it,
fat.
yeah, wow.
There's some fat brittle stars.
So much life.
And anemones on it as well. Thank you. Yeah.
That's so beautiful.
So it's really good. Okay. Just gonna need to open a little bit for the vessels in the way.
No worries.
Bridge RV. Need to go more in there. Yeah. Could you, I'm updating your waypoint now. Could you make that move at 0.5?
Yeah, 0.5.
Thank you.
This is nice.
Yeah, fantastic.
Wow, this is big. Look how it's
Absolutely.
not even small on there. Okay.
Yeah.
You think we could stay a little bit longer? Just,
Oh, really? Is that where we're going?
Before when we were in front.
When we zoom in on stuff.
You could get short of 20, 30 seconds like this and then zoom in 20, 30 seconds. Just,
Try yeah. Do you want it here?
great. It's beautiful.
Yeah, we can pause here.
Something
So-
bigger. The one earlier we measured around a meter.
It's pretty big as well.
Yeah.
Have we seen any actual seeps?
Are we saying sorry what?
I've got a feeling we see those get big shoulders.
Interesting.
It's not nice.
But we saw it from the middle or something.
Around, yes, but you know, listen, that is a great part.
Yeah.
It's just the slow move. It's
Yeah.
like a calm. Yeah. Might as well leave it slow.
This has all been behaving itself?
Eh?
This has all been behaving itself, yes,
Yeah, it's fine. We're not sitting on the bottom ledge, so the details.
Charlotte, you've seen this type of white coral before for the dive?
Thank you.
Not for this one. No. And maybe if we could just zoom in on any of them.
It's beautiful, right?
Yeah?
No, you got that.
Yeah.
Just ask them to have the back there.
Just gotta do water change.
It looks almost like snow.
Yeah, yeah.
Wow.
Oof.
That's really nice. Got some squat lobsters in and around it.
Would love to hear the sound of this.
And then of course on the rocks we've got sponges as well.
You may want to zoom in the sponges for it. Sponge people.
Oh, okay. Could we also please zoom in on this
You need
on
this
these
so it's
sponges
a
towards the
far back, or just, yeah, around this little baby rocks.
on the sponges, and then after we'll hold it for like 10 seconds. If possible, I know the currents are a little
Well,
On the flies.
can we
yeah
sit down?
on there. We're kind of like hovering above a.
I know, but if we can sit down, that'd be great. Yes.
Possible?
If I sit down, it's gonna be too up.
Too high?
Yeah. I'm going to park.
You're being Jumbo. Okay. Anything. Thank you very much.
And then if we could the white balancing, or maybe reduce the light so we could see the things a little better. Yeah, that's good. Yeah, perfect.
Wow.
Okay, so we'll hold here for a few seconds, and then, oh yeah, the base where the sponges are as well. So we've got a good few sponges. We've got like a benthic ctenophore towards the left, that red jelly-looking thing. that's perfect. Yeah. Amphipods as well.
That shrimp like trapped in this one.
Inside, right?
Yeah.
Yeah.
It's inside.
So I assume that's a carnivorous sponge.
Poor thing.
Oh, yeah.
Yeah.
Just right in the middle.
Yeah.
I think that's good. Yeah.
Yeah.
Thank you very much. Yeah.
See anything else, people?
Yes, but it's not here.
Is that a sea spider towards the back there?
Yeah. Or,
Well, it's called lobster, right?
Lobster? Oh. Yeah, squat lobster.
nice.
Yeah. That's good, thank you. Yeah, we're getting some good views. Oh, that's how your blue face also.
It's a beaver point.
Hmm.
Then it's kind of advanced.
we then heading straight ahead? Sunny?
What?
Are we then heading straight ahead?
Yeah.
Some more old dead corals here. Yeah. Is there? Oh, chimera?
Definitely the coral dive.
This is a nice one here. A coral and sponge bouquet, kinda. Lots of different types here. Nice. Brisingid. That's beautiful.
Where's your RV at the edge of the point?
So we're currently on the caldera. We reached the summit and we're kind of just going around the summit. Oh, what's that? To the left. Yeah. I don't think. Well, I personally haven't seen this kind of. Or, what is that? Anemone, yeah. Nice. What's that purple thing down there? I just that's true. Yeah. I guess I was just curious.
It's big, huh?
Yeah. It did look, it was blowing a bit, yeah. So throughout the area, we're seeing lots of bivalve shells and bacterial mats, which are all indicative of methane seepage. And then also on the hard substrates, the rocks that we're seeing all around, we've got lots of corals, lots of different types, and sponges growing on them. So quite a diverse area. So we're just keeping our eyes peeled for any bubbles coming from the seafloor. That'll be methane that's being emitted. Nice. Another rocky outcrop here. Quite a large one. So a mixture of dead and live corals. So we're seeing more shells again, and then this gray slash whitish mat that you're seeing on the seafloor will be bacterial mats. The discoloring that you're seeing. So these bacteria use the methane that's being emitted, and they transform that into energy, basically. So many of the organisms that you would see around methane seeps have a relationship with bacteria. They're either, within them themselves, like tube worms, or the crabs. We have plenty of crabs that feed on the bacterial mats as well. So lots of urchins in this area.
Yeah.
And then what is that? It's not just rock. Just a rock.
Bag.
Oh, it's trash. Uh,
Plastic bag.
yeah. Wrapped around, and then there's a bended Tina 4 on the top.
Yes.
Straight or go left? You wanna go to waypoint 6 or bubble?
I think
9?
bubble 9. Yeah.
Okay.
Well, you go to 6 and then to bubble.
Okay.
So 6
Well,
then?
6
Yeah,
9.
and then down.
Okay.
Okay. Bridge RV, new waypoint.
Another big outcrop. That's nice. Lots of wire coral. do a little zoom on that. Right there, please. Those like tube worms. Or... looks like tube worms. Right? Yeah. Got some live tube worms there as well. There.
A bit long. You can actually see a couple of tube sticking.
Right, some live ones, yeah. I mean, that's good. So, if you're just tuning in, my name is Kyle Foster. I'm a student at the University of the West Indies St. Augustine campus, studying deep sea biology. And today we are exploring a collapsed caldera-like mound. We found some pretty interesting things so far. Lots of corals, almost a coral garden basically. Personally, I think this is the most coral that we've seen for the entire expedition, so it's quite exciting. So we're on the caldera and we're just making our way around the slope. So we're close to the top, we crossed the top. And yeah, we're just making our way around. We've seen lots of rocky outcrops with plenty of corals. And yeah, so we're just keeping our eyes peeled for any signs of seepage or bubbles coming from the seafloor. Right. Yeah. So we've seen lots of bacterial mats and tube worms, which are indications of, you know, these are chemosynthetic organisms, so we've seen quite a few dead corals as well, scattered among the live ones. And a bit earlier in the dive, we saw like some of the biggest brittle stars I've seen just wrapped around this pinkish, purplish coral. And now I hand you over to my colleague, Shauna Edgel. Thanks so much, and you're in capable hands. To waypoint 6, and then we're gonna go down to the blue waypoint. You can just take that across a little bit. Yeah, so we're heading to 6, and then we're heading to 9 afterwards.
Okay.
So yeah, just
Is this okay?
I have
Yeah.
there's been some confusion between the two
Okay.
maps.
Okay, cool. Oh, you haven't done on the transects yet.
Transects, one collection, just looking out for bubbles basically.
Okay, cool.
Lots of coral in this area.
That's cool.
Yeah.
Nice. Yeah.
As you see,
That's okay. Hi everyone. So, as Cal probably mentioned, we are moving on to 8.6, looking for some seeps, looking for some bubbles and some high density areas. Fish. Fish, little bits of coral. It's like brittle stars on some coral. Or maybe they're crabs. sea urchin. Hmm?
A pretty rocky beginning, like rocks and boulders and it's
Well,
all turned into mud.
It's all charm. What is that? Is it? Can we get a zoom? Oh.
A bit of a nap.
He's mad.
Want some cream drops? I don't know. What kind of fish is it? What kind of fish is it?
Last time I asked, I was saw it. It's a very large flatbed, that's what.
That's
That is
huge.
That's not really very large. That's what I was told. You have the pictures there.
Yeah. I mean, six? Can I think you wanna?
Right next.
Yeah.
Off?
Yeah.
So we can watch it for a while.
Oh. Some shells. Mm-hmm. Couple more sea urchins. And a more thick ones, it seems. Oh, this one's really quick.
RV updated real quick.
Substrate's changing a little bit. See, it's getting a little more rocky. A little bit more interesting. See some shells.
Big seashells.
Oh, really? We can twist up and down. Forward. Wow.
Bridge RV, can you come to stop it?
Did you know? Something like that.
Bridge RV, could resume the move, please.
Mhm.
It's generally all.
You're getting a look from there, aren't you?
springs like this. High, but because it's gonna to go fast and it's gonna go
Some slackers.
could go backwards pretty slowly.
Okay.
What we doing? Sciencing? We cool?
You see, it's my back camera. I was touching the
Mhm.
ball.
Well, the blue one.
Blue
Blue.
bubbles?
Mhm.
We're going here, and then
This one.
that's yeah. Okay.
So, coming down.
Those things are cool. We saw one come down the other day. I've never seen you do this before.
Yeah, yeah.
So I don't want to.
That's what it was? It was like
Yeah.
sleeping or something.
Yeah.
You good?
Thank you.
I got you, boss. When we come up.
I'm guessing.
What's on the set? Layer?
Z2 Kangen Hans.
Or like, reflections or something.
I don't know how they are.
We have the Charlie and the portable lights and the box, but we didn't use it.
Lights on, right?
Yeah.
Doesn't lights all the time. But what about that outer? Is that facing on the south now?
Now we putting the porch on the side. We asked Johnny how he wanted, and he said that way.
Okay.
What else?
Different wax calipers. Calipers on each top side of the porch, right?
And Hollywood close to this now. Because we usually have the nets as well in the between the
Okay,
box.
we're on the porch. That's all yours.
Clean.
Perfect.
And have fun.
Got it? Yeah, almost there. Also, some other stuff here. Look here. There's two different
Yeah.
points because some of them are
Bubbles?
like they're drawing a bubbles. So over there is the blue ones are the bubbles. Here for some reason it's not going blue. Don't mess around because we tried them.
Oh, yeah? Double dip. Let's find the seat. Oh, sure. Michelle.
So also we have almost 110 because we start like doing days and then end like this. So it's bigger than it's a
Turn right.
yeah.
Gotcha. Interesting map there. Guten Morgen.
Good morning to everyone who's on the stream this morning. My name is Sharice and I'll be taking over for Shauna. Hi, good morning. I'll be taking over from Shauna now for the next hour as we continue exploring along this dive. We are currently at sites 6D which is a caldera-like mound that was mapped. If you guys could see this map right now, it is really cool geology that's happening. So I'm excited to explore with you guys, and we're heading along to waypoint 9 currently to look for anything that could be similar to bubbles, which were anomalous on the seabed mapping that occurred. Just to confirm that we won't have to go
Or before, yeah.
Maybe out some.
What number was that? I feel like every time we join each other, we're at a steep slope.
Or what?
Every time we join each other, we're at a steep slope.
Yep. Yep. Going down in the hole. That's what altitude is.
Ooh. We're getting some hard structure, which is always a good sign, and then a lot of like shells.
So, you know how to set that sonar up? What we've been doing? Try 100.
That's a chunky sea star.
now.
Oh yeah, we did go down into a bit of a dip, according to the map. Alright, so we're just gonna follow along the track. I think we're still gonna be hitting that point anyway.
Okay. So once I get to the blue dot, turn to 7?
Yeah.
Is that the blue track or that's something else?
I think it. Zoom out.
It's not quite. So I gotta go that way. Almost blue mark. Okay. Let's check.
You see shells at least.
Have y'all seen a bunch more of that coral rubble or no?
No.
No. That's so weird. Did y'all learn anything from looking at it really, or hard to say?
Did you look at it? Yeah. Sure is.
Oh, there's some. Speak of the devil.
It's fossilized coral.
Well, we don't know what it is. It's fossilized something.
Fossilized. I didn't also get a look at it either. I brought
Something
it
or
out.
other. Okay, I'm at the blue bubble.
Yeah, no, it can change.
Okay. So now I'm gonna point north and shoot north. Okay.
That's 7, yeah. Sounds good. I guess we.
Right now.
We haven't been to 7, have we?
We're
No.
heading to 7 now.
Yeah.
Should be right around this heading.
Oh, interesting.
Ready, gyro beat?
Alright.
A lot of
Take
shells.
it, mark your point.
A lot of shells.
Look at that chunk.
That is a chunk.
Chunk.
What is that? Xenophore, bacterial mass.
Bridge ROV.
Yeah, man.
Can you speed up the vessel to 0.4?
Yeah, we're at 0.4, so we'll increase to 0.5.
Okay, thank you.
Big rock. Whoa. Oh wow. Ready?
Good breakfast. Definitely a lot more rocks to my right. That would take us away from 7.
No, we'll keep on the heading. Right?
If you see some of these kind of small sponges that you think you could grab, it'd be great to get one. I think
Which one
these
exactly?
ones in particular probably look too on the rock, but like
Yeah.
I see one. Yeah, if you could grab that potentially.
Okay.
Where do you want it?
Do you want it in a silver?
Wherever you want. First two.
Okay. All right, so we've just had Rob, one of our sponge experts, come in, and
Bridge
You're
RV, can you
Niskin
stop the
first,
vessel, please?
yeah, we're gonna do the Niskin first.
You know, if your starboard arm is safe, you need a hand out.
That's one.
Yeah, you put it where we.
So we're gonna do a sponge collection. So with sponge collections, what you have to do is a Niskin first for water sample, and then we collect the sponge itself. So we'll do the Niskin first before we collect the sponge.
Sure.
First the Niskin, then the Niskin one. All right, perfect. All right, and then we put it into coral quiver too.
White hot dog.
How big do you want the whole thing or like a chunk?
I'd say just the whole thing.
Okay, the whole thing. It's hot that it's attached to, I don't have a,
This is close to the rocket.
yeah.
Towards us a little. Not on the rock. Not enough. Sorry. Just get resettled.
You want that sponge or not too?
Why? on to
Move on to
an
a
easier
different,
one.
easier sponge.
There will be more things.
Also, can we have a bigger sponge because this is too small and all that happens is you take the measurement.
Yeah.
Okay. Did you see one nearby since we took a Niskin one?
No, it's okay.
You want the ball.
When we see a bigger one, yeah.
Gonna take that one as well? No, this is too small. We'll go for a different.
Yeah, so like a lot of these encrusting ones on the rock, it's pretty easy. If you see ones like coming out, like up and up, they just hit me.
Okay. Sounds good.
Yeah, some bigger.
See if there's any. And just take a new Niskin. Take a new Niskin, yeah.
Step bridge move again. Bridge ROV,
Go ahead.
can you resume the move?
Is that one of that's okay?
That one's gonna be really hard to do.
Okay, no.
Up, down, and roll. Valley.
Is that one?
Can't tell if it's any bigger, but
Try to get a sponge that many people can use.
Mm-hmm.
if it's
So
so big.
big.
Like not like plastic. A lot of little white balls that we've seen around.
Look at that. Looking for a huge one. Some huge boulders, no?
Yeah, that's some big ones. Oh,
Hmm.
there.
What? You see something?
You see. I don't know what I see. It's dirty red. Oh, it's just the rock, sorry. It's hallucinating.
Massive.
Yeah. Also that's small.
So there were probably sponges around
Wow.
What is that? It's a weird looking one, huh?
The yellow one?
Yeah.
Something.
So what?
Looks like a transect.
Atlantis.
Yeah.
Oh, look at that fish.
Holy, that's a shark,
Shark?
huh?
We got a shark.
It's a deep water shark.
Wow.
Can we get
Do I have to call up?
Yeah, maybe.
Oh, don't come at me, bro.
Pictures. You're spamming screen grab.
Is there a number?
380, I think, is Usha's room.
1383.
What a find.
Stop coming at me.
Yeah.
I don't want to be friends.
It's been hit on the head.
It has been.
What do you mean?
Hi there,
They
this
already
is
got some
Anna. We
blood.
have I think a cat shark on the video.
It's like that, my friend.
Interested?
Follow the camera.
Well, no, we find
no.
it down here,
No,
but
there you go.
she's close.
Don't do it. Don't you do it. This guy's
You
huge, huh?
can, yeah, just put it on the.
Huge.
Is it a cat shark? Looks like.
Oh, he's got weird face, man. Bomb. Kind of cute.
It is.
It's like a Pokemon.
Oh, look at the face.
Hello.
What's pets? I can't give you pets.
Shark. He's smiling. I just told Lucia that she's gonna check. Did she tell you? Okay. She's awake. I just told her about this.
I'm not sure how long this dude will stick around, though. Just FYI.
Tell me.
Now might be your opportunity.
It's a friendly one. Right. Yeah.
Right, so we're back on the regular camera and we're heading back down to the bottom. We about 30 meters off bottom. After we followed that shark for a bit, which was really nice to see. That was so cool.
That was really cool. I didn't even get bit once.
Yeah. That moment when he just like stared at you was cool.
Still got all your fingers. Like, what is wrong with you, dude? Very cat-like eyes, huh? I feel like the spiracles are diagnostic. Alright, birds can start moving in if they're not already. Right there, Opie.
Yep.
Are you still moving, right? That's what
I had stopped, but I'm moving again.
Copy.
Okay. Still on the lookout for the sponge. A lot of shells.
Bacterial matting.
Mm-hmm.
Definitely a lot going on here, huh?
Indeed. Somewhere, a lot was happening. We're climbing again.
What's that?
We're climbing again.
Yeah. Back up this top of this ridge here. More rocks coming. Shells coming up.
Mm-hmm.
Creeping over a little bit that way.
There's a lot of shells. That's a lot of dead clam shells, yeah.
Seems like we never see any living clams.
Well, they're usually buried beneath them.
Very How many do you think are fluid beds?
So if you see a little bulbous sponge or something.
Yeah, just let me know if you see one. I haven't seen too many yet.
Sponges all the time this morning.
There's like a low sponge area here.
More bacterial mounds coming up too. A lot of shells.
Got some somewhat interesting sonar hits out by waypoint 7. That's not an expected bubble location though, is it?
The dorsals look different in
I
the
don't.
illustrations, but
Maybe it's a what?
I don't know, we're still figuring it out. Yeah. And then we got a good video of it small, be able to go, it's this thing. Taylor's not place. Taylor's not.
Yeah, the tail is not as.
Bubbles at 8.
Yeah.
We'll still head to 7 and then change heading to 8.
Sure. That sonar hits like just 10 meters earlier than 7, so if you want to just get whacked at and then turn and go over 7.
Okay.
Just to see what it is.
Sure. Sounds good.
There's big rocks going north.
I mean,
Who
big
knows?
rocks means there's no like change of flight.
Sorry?
He said there's a sonar hit for something big, so we're just diverting slightly to see if it's like rock structures, and then it's on the way to 7.
Yeah, it's like 5, maybe 5 meters, 10 meters. In there. Maybe buried one.
Buried starfish. For anyone now joining in, we are currently exploring Site 6D off of Trinidad and Tobago's waters. It is, which is located in the Atlantic. We would have seen a shark earlier, and currently we're heading towards waypoint 7 of our track. So far, we've seen a good bit of coral, some sponges, a lot of clam shells, which
Yeah. Once you get there, then we're going to start moving you to the northwest, Al. Okay, so.
yeah, so we've seen some rubble activity, some rocky outcrop areas,
Maybe, yeah, that's fine.
and we're just continuing along
Maybe a little
on
more
this.
north of the line, like this. Just so I can kind of get beside him, you know what I mean? Instead of him pulling me forward, it's usually not great if he's pulling me forward. What, 7?
Tell
Yep.
Tony,
It's like everything is like this little portion doesn't have that.
You can start moving. Keep moving now. Roger, 0.4.
yeah, trying.
Ciao.
That's probably not gonna happen, but I would love to find patches of some holes that's not visible. You know? See what it looks like. Ooh, something big. Big and rocky.
How far out does this thing go?
Right now, I think we're set to 100 meters. You can set it farther, but it starts getting pretty
No,
noisy,
yeah.
you know? 100 meters is usually plenty for this. You might be able to see bubbles with it, but you'd have to stop, come high up enough off the seabed that you're not getting returns, and then probably scan super slow. Probably do like a 10-minute thing, you know? You don't want to scan 100 meters of thing.
A lot more shells coming up. Makes me wonder how many living clams we've passed over.
Yeah, I wonder, are they like the coral, or are they mostly all dead, or are they still around?
No, they're still around, just pretty buried.
It's all in like rows. Stuff that's still standing there.
Ooh. Where are the sponges?
That's what
Something.
Farah says.
A bull cat shark?
False.
False cat shark.
Oh.
It is funny how you left sediment on it.
Look. This is the false cat shark.
Oh,
Go for it. Watch out.
that looks it. That looks like
That's
it.
it.
Is he really big?
Yeah, like that.
Yeah, that's what it looks like.
Yeah.
Oh, especially head on.
But look at the fin.
Yeah.
There's a long
Yeah, the
It's
dorsal.
got this
dorsal and then a long tail.
Yeah.
Really big. Silver shark.
A silver shark. Okay. I like it. Soul member was Chinese. Where's he
I stomp around all those three clusters there.
Yeah.
So just set them up just to the north of there where I can not go on the red bubble and see them all. Oh, let me.
Sorry, Jeremy.
Oh, T-berms.
Update the waypoint. Here we go.
Why we get excited about seeing T-berms is that they are a sign of methane activity or methane seeps. So when we see those guys, we start knowing that we're getting close to summer bubbles.
We got 3 dots all right next to each other.
Yeah, because it's waypoint 8, which was put right in the middle of the 2, so we'd explore.
That means it's going to be 3 times more often, right?
Fingers crossed.
Alright, so start with 10 and then 8 and then 11.
Still keeping an eye out for sponges for Rob.
That's a big hole there. Rocks are getting pretty interesting.
Mm-hmm.
Can make that biogenic stuff, huh?
Oh wow, that's a lot of shells.
A lot of shells, a lot of worm, column casings. One close at a time. Hey Alex, can you zoom in on Fruiter Mouse too, or 40 nav or whatever that is? Just make sure these waypoints line up. I'm doing it right. Might have to shrink there. Yeah, Titanic size ROV might have to. Right over 10 now.
Right.
Vertically. Heading straight for 8 and then 11.
did. We went down to 9, and then we now went back. Then we went back to 7, and then 8. So I don't exactly know what path we're gonna follow. But I think
And from 9 to 7 and
yeah. So I think 9 was actually a lot more down than anticipated. So we ended up just doing that. And then 7, now we're at 8, going along the bubble hunting route, and then if we find nothing, just continue. We haven't taken any samples except the attempted spend. But we're all good to go. Alright, bye guys. I'm gonna hand over to Rob, and thank you so much to the pilot.
How about the second side?
Hi everyone, Robert Cassidy here, PhD student at the National Museum of Natural Sciences in Madrid, Spain. Taking over for Charisse on this episode of Bubble Hunters.
Cassidy.
How you doing?
How you doing?
Good. Cool shark before.
Yeah, that was cool, right?
Yeah.
Alright, back up to 8.
That's good. And if we were at 9 already, do you want to just go in the direct path to 10?
Where are they?
Yeah, she said that we went from 9 to 7 to 8.
There's bubbles, right? There's the names of the bubbles and there's the names of the waypoints. So things should just very.
She was,
We haven't done that, have we?
she was saying we went from 9 to 7 to 8, but no.
No. I don't think so. We did.
I'm
So
just
there's-
getting to 8 now.
Yeah. So there's 2 types of waypoints.
Okay.
One is the bubbles that have this different numbers. So
Yeah.
maybe should
Okay.
We've done 10 and 11 bubbles. Is that what she's talking about? Did
She just
say
told
that-
me 9 to 7 to 8 is what we did, so maybe there's some confusion. But yeah, I'll just keep going then.
We haven't done that.
Normal track.
It's not that.
Yeah.
Alright, and the 9.
Yep. Sounds good.
For the next dives too, if you want, we could make the bubbles and the waypoints all one line.
Okay. Yeah, I'll see what we say. What Anna and Viva think.
Okay. Trucking. Put a new move if you want there.
Reach ROV, update the waypoint.
Just in time for your watch, man.
Perfect.
Just feel like
Yeah.
skate. You care about skates or no?
Yeah, we get red skates. Can we zoom on that, please?
Yeah. Sure can.
Eyes, spines, and everything. Yeah, so we want to try to get as much of a good zoom as we can. First, I'd say the whole body, and then we could zoom on like the eyes, and then try to get these spines if we can from an angle. So first, we can just have it on the whole organism. Thanks.
Okay.
I think that's good. That's some good
Closer?
grabs. Yeah. Sorry, and then I want to do a closer one of the eyes also.
Sure. A little closer.
Yeah.
I'm trying to sit down.
So we've got another skate here. I'm not really a skate expert by any means, but it seems pretty similar to some other ones that we've seen on this trip so far. And what we're looking for, oh, spooked him a bit. We're looking closely at the kind of configuration of the eyes, as well as the spines on the tail, and various other characteristics, just to get a good visual ID. Some good images for our people who know more about skates, like Bea. Varnasp. Yeah. Yeah, and that's good. I think that's probably good for the zoom. Okay.
Roger, Roger.
Cool. And keep moving on towards the 0.9. Other fish down here. We've been passing through a lot of pretty rocky and kind of sloped areas today. Coming on in like a little bit of a flatter part now, but we are continuing to explore the area around this kind of caldera-like mound, where we believe, well, so the site was mapped before in the 80s, and it seems like either some of the initial mound has collapsed since then, or the mapping data was not initially as good as we have it now. So we're just continuing to kind of check out the different areas of this mound, looking for seeps, trying to get samples of any new fauna that we find. What have we got here?
Deep sea isopod.
Big isopod. Wow.
Wow, let's follow him for a while.
Yep.
Yeah.
Do you care about the isopod? Yeah. I'm talking to John.
I care about the
It's a nice
isopod.
giant.
These are huge isopods. Just look at the scale bar for reference. I mean,
It's not a pill bug.
yeah. Looks kind of like a roly-poly, but these are, like football sized.
Just for us. not for the-
Just for us.
Oh, he's fast.
He's fast. I
Yeah.
didn't realize they're that fast.
I think it's spooked in the room.
So we've not seen many isopods with the ROV so far, but we have seen them on our Doris cameras, where we have when we've dropped them on this trip, we've had these bait cameras where we have some fish as bait, and then we get kind of different images over the course of a long period, where we get different scavengers, and these are really good images because they don't have like the loud sound and lights of the ROV, so you get different fauna, and it seems like it's easier to get isopods in those. Wow. You just yeah. He was really fast, but now you're just taking a little break. Faster than the shark before. Wow. Very cool. And he's off again. We're in hot pursuit though. Oh, okay. Yeah.
You said catch him?
No.
Okay.
Catch up with it.
Yeah.
Say, what are you gonna do with that?
is too much for one person to eat.
Yeah. Ah, he keeps bumping into the sea floor.
Super cute.
Yeah.
You move the boat a little north. Super smart. He's trying to get me to go into the circle of
Bridge
death.
ROV, can you move the vessel to the right?
It's very prehistoric, didn't he?
Yeah, very much. Yeah.
Wow, Eugenia, those are great shots.
Yeah. Wow.
Great shots.
I know. Yeah. He's doing some kind of evasive maneuvers right now. I mean, pretty amazing to see how it swims so high up off the floor. I mean, usually you just see these things kind of skirting along the bottom. That's so cool. And look at those little legs just kind of moving it along.
They're like propellers.
Yeah. I mean, it's amazing because it seems like it would be too dense to really swim that well, you know? Wow.
Yeah.
Look at that.
But it looks like Al's evading.
Also doing evasive maneuvers. That was pretty good.
Oh.
Ooh. I might have stirred up the viz on that one. I don't want to hurt you, buddy. We're just looking. We're just tourists.
Wow. It's pretty amazing that something that armored can move so quickly. So cool. Yeah.
Little swimming flaps. Swimming flaps, I love it.
Wow.
So fast.
Yeah. So cool. This has got to be one of the more charismatic isopods out there, I think. Very cool.
Well, Eugenia, you caught a shot where you can actually see the feet.
Yeah.
Wow, amazing.
Oh, you guys had about enough of this dude?
Yeah,
Or?
and he probably has had enough of us.
Yeah, I think so.
All that big meal that he had, he just wasted it.
Yeah. Got some exercise. Yeah, I think we can keep moving on with this guy.
Let him go about his life.
Yeah. Go back to his business.
All right, little dude. We love you. Good luck in life. About to swim on out of there. That's actually my first one of those.
Really?
Me too.
Bucket list.
First one swimming.
It's a bucket list guy.
Yeah.
That was about the
And we saw a little bit of footage of them swimming off on the Doris, but nothing like that.
That was nice enough. He actually helped me take a turn out of the umbilical, too.
Yeah, where are we now?
We didn't actually go too far
Not
off.
too far.
Yeah.
All right, Al, I think we're done bug chasing. Sorry about that. Could you see that up there?
I could. Good thing.
Yeah, that guy was crazy.
Now we're back to our sandy sedimenty exploration.
Good evasive action on there, Al. Sure. Yeah. Is that the same dude? No, that's
That is a cuke. Yep. Oh, another swimming cucumber.
Somebody must have been hauling butt through here. Look at that.
Yeah. Wow.
You can come a little further south, so let's do like
Over towards.
something like that, okay?
Supposed to go with it, it's not going that way.
What's that?
This is the video transects. That was the dive plants there.
Sorry, I can't hear you still.
Video transects.
Oh, you want to do it now?
Well,
Well,
supposed to. Oh, it was supposed to be 5, 6, 7.
haven't done any. Yeah, we can do one here maybe.
We can do one after 9.
After 9. Okay.
So we got.
Yeah.
Just take
The
box.
issue is that I keep taking like video transects when it's like pouring sediment. That's gonna bias the transects, right?
Yes.
If we only do them when we get
So we're starting at 9 and we're going.
objective decision.
Luckily, 9 looks quite boring too.
Good.
Do
Do-
we,
300 meters.
each other? 100 meters each segment.
Each segment is 100 meters per total.
Exactly what they say.
Sunday. Bro, there's a lot of cool stuff here. Did you see that shark?
No, I didn't.
Did
See the isopod?
Yeah, I saw what you were just chasing.
That was on my deep sea buckles.
They do.
It was it?
Yeah, we checked a few things off the like in the deep sea
I
buckles.
took
Yeah,
a turn out of the
okay.
umbilical while I was following it.
Eel.
Why you what?
While I was following
Get
it.
my swim.
Because it went. whoop.
That's
it.
That's pretty amazing.
All right, so yeah, I think we're gonna do a transect starting at 9. It's been alternating between this and then really cool.
Okay.
So. Roger, Roger.
Go have some eggs.
They got eggs?
I got it.
Okay.
So you stop transit from the station,
Yeah, from 9.
Okay.
It's gonna be 300 meters. So we've got a pretty calm stretch right now, but in general, this site that we've been at for the last 2 days has been very interesting. In addition to this kind of, the shark and the isopod that we just saw a minute ago, we've also found a lot of sponges and a lot of interesting kind of fossilized rubble that we found on a slope yesterday, I think, which we think are fossilized tubes of tomb worms. But we have not confirmed that yet, and we've got some samples hoping to do some radiocarbon work, I think, on them to get a better idea of how old they might be and what they might be.
This case around measures 3 or we do the third?
So
Because that would
we're starting at 9,
that would be the third one.
so it's gonna be like 1, 2, and 3.
Yeah, that would be the third one.
Yeah.
What do you mean the
What
third
do you?
To
3 times 100 meters. That would be the third 100-meter. The
That's
third.
the end of the.
That's the end of
But
it.
the problem is there's a turn on the line.
I
Is
see.
that straight?
No, turn. You need to turn on our line, the track that we.
100 I didn't compute.
The 9 to 10 is not enough. Is that what you're saying?
Yes,
We'll stop at 2.
Just to do two.
Yeah. Let's start on it. It's a swimming
Thanks.
sequence under Igneous.
What's the genus? Did you say the genus for this one? Okay. So that was an Enipniastis?
Probably Xigria? I don't know.
Yeah. Leave it at Enipniastis because that's what I can pronounce. Also known as a headless chicken monster, I think, right? All, yeah.
You marked it already, right?
That is your first.
Okay. Then I totally like.
So,
Yeah, like.
so we're starting now. Yeah, so starting a video transect now, which we do periodically to have some standardized visual data from these areas. We're moving at a constant rate here, about 0.1 to 0.2 meters per second the whole time, and staying just a few meters off the seafloor. 1 meter, sorry. Yeah, 1 to 2 meters. You could go just a little bit slower. That just says. Oh, what?
That's fine.
Okay.
Decided 0.1 is this.
Oh, we said 0.1 is just okay. You said there were no transects so far, right? right here.
Jeremy.
Yeah, go ahead, RV.
Good morning. Just updated your waypoint.
Morning, Mike. Thanks for letting me know. I'll head over there now. Cheers.
First 100 meters down.
Got a few of these sea urchins here. I believe these sacs that you can kind of see among the spines are venom sacs. We have collected one of these so far in the trip. You see them quite a bit in these more sedimented areas that we've been sampling. Little fish here as well. Very pretty ctenophore, looks like. What's that? Yep. Ctenophores are pretty interesting because they look a lot like jellyfish, but they're actually a completely different phylum. And they're also interesting because there's, among the evolutionary biologists who study early lineages of animals, there's a pretty big debate about whether sea sponges or ctenophores are the oldest living lineage of animals. Every few years there's a new paper out that uses a different kind of genomic technique, and it looks at a different set of taxa from these different phyla and concludes that they definitely know now that it's either the sea sponge or the ctenophore that came first. I personally am in favor of sponges. Of course I am. I think I'd be fired if I weren't. And that's because, well, you see, so there's all kinds of different things here. But if you just look at the morphological characteristics of sea sponges versus ctenophores, ctenophores have a more organized body plan that's more similar to later lineages. And they also have like a very, very rudimentary nervous system, right? And these two things are like big components of animals that sponges lack. So if you just look at morphological characteristics, you know, sponges definitely seem like they'd be the sister lineage to the rest of the animals. But of course, it's more complicated than that. I won't get too into it. Yeah. It's an interesting circular kind of pockmarked area there. Okay, so that's the end of the video transect.
No, still have to get them.
But don't have another one, so.
But that was the end of the transect, right?
Uh,
That was 200 meters?
yeah.
Yep. Great.
So that was the end of it?
That was the end of the video transect, yep.
Well, you want me to mark?
No, that's fine.
That's okay. Thanks. So that's the end of our transect. Seen some tube worms now, so maybe some bacterial mat here as well, just a little bit. Those are signs of seeps. So, there's a chance we're coming up on another seep, or maybe just a kind of mini seep-ish type place, if you're lucky. Looking for whatever we can get, really.
There's the dog.
Oh yeah, that one's alive, definitely. And now we're seeing some clam beds, okay. So these clam shells are also usually signs of seeps, sometimes active seeps and sometimes kind of dormant or dead seeps. Keeping an eye on the sonar too to see what we find. I see some rockier areas over there. Maybe we could kind of do a little flyby here. That's all right. Yep, some more bacterial mat. This white mat here is definitely a seep sign.
That's the rose.
What's that floating over there? That's coral. Wow. That's a cool coral. Do we want any of this coral? It's like when we saw the other day. You don't have it. So maybe a snip? Yeah, it'd be great to get a sample here. I think just a snip of one of these branches would be good. Whatever you think is easiest. We don't need that much. Maybe just like 10 centimeters.
We're gonna stop here for Thank you.
So this is looks to be the same kind of coral that we saw the other day when we were exploring the fossilized ruins on the other side of this large mound.
Yeah, got about 5 meters.
So we're gonna get a sample here. Yeah.
I'll lock it down and see. Okay. So where do you want it?
I think we can do Coral Quiver 2.
Okay. Did you? Yeah.
It's a good from the base, a good images first.
Yeah. Are you saying you want the whole thing?
How big it is?
One branch.
Yeah, for the branch.
Just a
But
branch,
let's took
yeah.
some good
Yeah.
imagery first.
Oh, we take some
Yep.
pictures first.
Oh,
If we could get a zoom on those like polyps close up, that'd be great. Just make sure that we're getting some good snapshots, please. Oh wow. Ah, it's beautiful. It's amazing how kind of like plant-like but yet alien some of these corals seem. This one to me looks a lot like a monstera plant that I have in my apartment. But it's got such amazing polyps. This is a black coral. Wow. Just that one little rock is all it needs to stay planted here. Pretty amazing.
so if you can try to get some zooms to the base of the coral.
To the base? Okay.
Yeah, you go. It's faster, not to.
What's that?
The
Oh, he's, oh,
focus.
okay.
Let me try the auto with my.
Oh, there we go. Wow. So that's the base of the coral. Is that very good? Okay, so I think we're good to sample when you guys are ready.
Okay. Close? No, too much.
How hard is their throat?
What's that?
I think it's loose.
Yeah, close there. No. There you go.
Nice. Mm. Do you want to risk it if you need the whole thing, or what do you think? Another one that's on a, yeah. I think we can abort this one then.
Release.
Yeah. Yeah, if we see another one that is on a bigger rock that won't move, it'll be easier. So no sample. Okay, so we're not sampling this one, but it has been nice to have a closer look for sure. So I think we can just, yeah, keep moving on track.
What do you need, Rohit?
Hey, Mike.
Red just like to move again. Thank you.
There might be a sponge on the right. Just fly by. Can't tell. I don't think so. No, it's just an old clamshell. That's okay. So. This again might be a sponge, might be. Oh yeah. I think we should try to sample that maybe. Been looking for a sponge all day. So I'd say for this one, it's probably too small to get just a piece, right? What do you think?
Yeah, it looks small. You could probably take the end out.
I think just take the whole thing. Yeah, and we could do some BB3A, I'd say. So this looks like a glass sponge, maybe something similar to the Hyalonema sponges that we've been seeing a lot of on this trip. A little bit different though. It's not stalked, but it does have this kind of like tulip shape to it a bit.
Do you want the rock it's connected to? Or
Sure. Looks like a shell to me. Yeah, we could take that.
Morning, Gerald.
Yeah, ROV, great. Good morning.
Good morning. Are we going to stop here for a sample, please?
Okay, let me know.
Yep. Yeah, so first we're gonna get a Niskin. Oh. Okay, I'm being told we don't need a sample of the sponge actually.
they don't need a sample anymore.
Oh.
So move on.
Oh, we can stay zoomed for just 1 minute. Sorry, just gonna stay zoomed for a little bit longer while you figure out. Yeah.
What's your plan?
So, sure, saying we don't need it, but I think it'd be good for DNA.
Okay,
I'm
yeah.
sure what the reason is.
Yeah, Megan was telling me the same, but I think it's tiny, so you will not be able to take a piece.
I mean,
But yeah, I mean,
for a
even if
piece, yeah. Yeah, I think we can sample this. So yeah, just what we said before. If we could get a Niskin before we start moving the arm, that'd be good. Do you know what this circle pockmarked impressions is? No?
Okay, too far.
What do you want, that rod, that Tobago, or the connector with the mesh?
Biobox.
Biobox.
Biobox is good. Yeah. I mean, if we could try to take just a piece, it'd be great. But, yeah. Yeah.
Whole thing together.
Do you think you can do a piece? Too small?
Yeah, most likely it'll destroy.
Because if
Yeah.
you put the jaw half,
Right.
the size almost of more than half.
Yeah.
So yeah,
Okay.
be willing to take the entire.
Do you think it's fine? I mean, just take it. Yeah, we'll just do the whole thing.
Whole thing. Take the shallow rock or whatever.
Yeah. Thanks. Sorry for the confusion. Yeah, this seems different than the ones early on, right? It looks like Hylianema similar, but not like quite the same. I thought.
Yeah, I mean, we collected a couple that were not stocked.
Yeah.
So, but we can also go.
This one just seemed like pretty different from the non-stock ones that was on the board.
Yeah, I
Like
mean,
that big one.
if we get the whole thing, then we can just spread it in.
Yeah. That's good.
Mama shop.
Yeah. Thank you. So we're gonna just pause for a sec to get some photos.
Yeah. Perfect.
Great. Yeah, I think that's good. Can you tell me where you are sample number? Do you have one for 6-7? 467?
The next one was 469.
Okay.
Right.
And 468 is in Niskin, right?
469.
Okay.
But 468 was in Niskin.
Was one, and then two is 469, and then this one is 470.
Okay, that's what I was asking. Thanks.
Great, then.
Well, which box is it?
It's BB3A.
B.
B.
Yeah.
Thanks. That was Niskin 2 that you guys fired, right?
Yeah.
Thanks. Great. Thanks, guys.
What are you, Jeremy?
Yeah, go ahead.
Can you resume the move, please?
I'm only resuming now.
Alright, so we're gonna pass you all over to Zahir.
So, yeah, just have these 4 samples, Niskins, coral here, and this bunch here. This is nothing. So, maybe ask about, actually, if they did a transfer at some point, with one eye, but no, they didn't before. So,
It's okay.
We're on, we're going between 10 and 11 right now.
Yes.
Morning, everybody. My name is Zahir, and I'll be with you for the next hour of our dive. So currently we're transiting between waypoints 10 and heading to waypoint 11. Continuing to conduct our visual surveys, keeping an eye out for any interesting features, possible bubbles that indicate seep sites. See a black coral off to the left, and a couple of shrimp, 2 different species. Pardon? Yeah, they think it worked real good. Easiest so far, yeah.
Trying to design on your just one using one lever.
Needing one.
Just using one lever and one arm.
Mm.
Taking me too long to try to design. So it's gonna make the easiest thing. Give for at least a week and I'll try to design it.
But it working. It working well. That was the sticking point.
We have a lot of time because we're almost there, so when we see something,
Stop and take a look.
yeah, just see if it's something that we don't have or yeah.
If we see something, we take a look, yeah?
Yeah, see what.
We have some time. So on our dives, we do take something called push cores, where we take a cylinder and we insert it into the sediment and extract out a column of sediment. So one of our RV pilots has also has a background in engineering, and we've been struggling to get the push cords out of, or the sample out of the push cords, and I think he has been constantly redesigning the push core. This version is up to 22, I think, but the latest iteration has been working rather well. So not only are we trying to evolve as scientists, but also as engineers. And this particular ROV pilot is actually from Trinidad, so another anecdote there. So we're just casting around to see if we see anything other than sea cucumber trails. A couple nice shots of this fish. Rat-tailed fish, probably. And the ever-present sargassum. The squirrel here. So we're gonna try to sample this coral that's under the ROV.
ROV. We stop the movement, we're gonna do a sample here.
Stop it now.
Yeah, it's on the logo. Yeah, that was 10.
ROV.
Hey, go ahead.
Sorry, buddy, may you come back about 20 meters closer?
Oh, no problem.
That's helpful.
So we're just gonna wait till the turbidity settles a little bit, and we're gonna pull back just a little bit to get some space to sample this coral. And welcome to our viewers from all over the world. Happy to have you all. feel free to post your comments and questions in the chat.
This one seems smaller, right? That's one. Then we can take the whole thing.
Okay.
On the base. The other one was to do.
Oh, when it does clear up here, take some shots before. Next number should be 71 for 71.
Yeah, for them.
Turbidity is making focusing the camera a little bit difficult. Click on the lasers, please. We took, okay. All right, let's get the image back.
So what we could do, we could, since we're already here, pick it up and then do the land trucks.
Yeah, if it's not clear enough, go ahead now.
And current is coming from behind us, so they're pushing everything in front of us. So the reposition, we'll stir up the vis again and take us a little longer so we could sample it there and take pictures higher.
All right, we'll get some pictures here though. Yeah. So we're gonna try to take this pink, beautiful pink branching coral, and possibly its squat lobster associate. So we're gonna try to take the entire thing because it's a very small sample specimen. So we're using the left arm. This arm does have a blade attached to one of the jaws to help us with our snips. So we're gonna get some shots of it, some frame grabs here as well.
Okay, that's good.
Ah, there we go. Yeah, we're good to go. Coral quiver 2.
Okay.
So those white vessels you see are coral quivers. It's what we put our coral specimens when we sample. helps to keep them upright and rigid and safe on the journey back up. We have a lot of turbidity, which is making focusing the camera extremely difficult. But we'll work through this.
Okay.
Yeah. You want to go in the large one?
Yeah, because I'm afraid of the damage in there. No, don't need to get the large one. Just don't want to damage the brass.
Okay. So changing tact here a little bit, we're going to go in the bio box instead. We've got a lot of current that may take the specimen away as we open the jaws. So the porch will come out. We'll go 3A if you want. 3A. Yeah, it's empty. 3A. Nice. So we've successfully deposited our specimen in biobox 3A. So we'll just reset and continue along to waypoint 11, casting about for anything interesting.
What do you do here?
Yeah, go ahead.
Resuming the move.
Okay.
Thank you.
So we're currently on top of a ridge on the edge of the what we're calling an ancient caldera. A likely spot for seeps and its associated biodiversity. So we're just going to explore the top of the ridge as we've been doing all morning. A bivalve shell there with a satanophore possibly growing on it. We saw a really interesting gulper eel yesterday. Euripharynx pelicanoides. Interesting name, but fitting if you think about it. It's often said that the mouth is bigger than the fish's stomach. It has one of the largest mouths relative to body size of any fish. And if you go back to our stream yesterday, when you get a chance, you'll be able to see this amazing creature that we saw, the gulper eel. As I said, its mouth can open very, very wide, much like a pelican's pouch, giving rise to its common name. Despite its enormous mouth, its teeth are actually very tiny and can't chew. It's a strategy in a food-poor environment. Food down here at the deep is very scarce and quite unpredictable.
So does it eat bigger things on itself like the black swallow?
Say it again?
The gulper eel, does it eat larger animals on itself like the other one, the black swallow?
No, I think its mouth can open bigger, but the swallow would eat almost anything. You know? But it does open quite large.
So what does it feed on?
Basically anything it could get its mouth around. Other fishes, crustaceans, a lot. But it's actually not an eel, it's a fish. And if you look at the tail actually kind of glows. There's some bioluminescence in the tail as well. Maybe for luring prey into, like some of the other anglerfishes that use bioluminescence as a way to lure prey in this very dark environment. Well, eels are fishes, but they have different morphology, different strategies in mating.
There you go ahead. Got it. Some black coral.
Yes, we can see a bunch of them. But
Take?
we have no,
We
some
have plenty.
photos.
Yeah. Okay. So we've already collected some of these specimens, so we're gonna leave them be and continue along.
What's here on the left side? It seems a bit different.
You wanna pan to the left a little bit?
Yes.
We do have a little feature on the left here, and something up in the distance. A rubble field, possibly some fossilized tubes. Satanophore. So, as I was saying, eels are fishes. They're just a specialized category of fishes. They tend to have little or no scales, more elongated bodies, and they swim by undulation. And our gulper eel can, as I said, open its mouth quite large, similar to the black swallower, which can also open its jaws very, very wide. Oh, it's an interesting one.
Some details of this.
Pardon?
Some details of this.
Stop the vessel, huh?
Will you stop the movement, please?
Come if you'll stop.
Let's see what it is. Wow.
Is it? Yeah.
It's a
You think
sponge.
I think it's a ping pong.
No, you haven't seen this one. Wait, she will come learn.
Is it the pomponi?
We haven't seen this one.
Mm.
I've never